Yeah, we're running a little over time, so we'll need to withhold questions in the next couple of presentations till they end, and then we'll have the group question answer session. Thank you. Thank you, Dr. Schnecke. It's very interesting. And so the next speaker, Dr. Marina Emborg from the University of Wisconsin, will be speaking about non-human primates.
Not coming through. Okay, let me try it again. Sorry about that. You know, when you have a nightmare that things are not going to work, I brought 2 flash drives. Neither of them worked, and they were working in the hotel. And now this is not working. Come on. It's working in the computer. What you call? Okay, it was working before. It was working before. I tried it. It's already set up. It's already set up.
He's coming now.
Okay. I can start because we are going a little late. So the reason I was, first of all, I want to thank you for inviting me. And I want you to kind of change the hat that you have been wearing until now, thinking about making transgenic monkeys, but put it from the other point of view. Imagine you are a scientist that does preclinical research, and you're trying to find a cure for Parkinson's disease. That's me. You could change that with your disease of choice. Can be cancer, can be diabetes, can be leukemia. And let's see if we can make this work. Is it in the right?
Okay, let me do that over there.
Okay, and my nightmare is just perfect, huh? So, so what I want to talk to you is about this perspective from a preclinical scientist, and I put a lot of, yay! Thank you. I'm done now. So this is me, and this is the first thing. So I spent a lot of, because of my research in preclinical applications and clinical research towards the clinical translation, I spent a lot of time thinking about the animal models and the way that we use them, how they impact the clinical translation, and how important is the quality of the research that we're doing from the moment that we start working with the cells and we are defining which compound or what genus of interest, going to the zebrafish to say, okay, maybe this compounds we are screening until we get to the rodent, we get to the monkey. Each one has a very important step. And when we're thinking about the applications of genomic editing, it being non-human primates and other species, it's not just making models and that we can do it by putting on a dish and doing genomic editing in somatic cells, but also creating a monkey that has the expression in the germline. But it's also about the possibility of creating therapies with this technology. So before we're thinking about applying genomic editing on human primates, let's think about why do we do research on human primates? If we have this wonderful models, I hear there's a lot of in vitro models, why do we need research on human primates? Well, we need them to understand the disease. We need the system because we are not just a few cells working in isolation. We are a complex system. And because we need to test new therapies, first-in-class and basic therapies are the typical target therapies that we test in non-human primates. Why? If you work in Parkinson's disease, you, for example, want to know how the fine motor skill is affected and if the therapy that you're using is being helping those patients. And monkeys are a better model than a rodent or a pig. The same way it happened when you're thinking about executive planning. And then has to do with the complexity of their brain, of their overall molecular, not just the sheer size. And what you are seeing here, okay? What you are seeing here is this is side by side. This is a coronal section of a rhesus monkey brain, and this is a coronal section of a mouse brain. And it's not just the different in size, but you can see there are the different in the structures. Here is the caudate and the putamen that the mice have it all together. So this is very important when, for example, we are delivering something into the brain. We can do, just like humans, we can do MRI targeting and assess the distribution of molecules. So this is the reason we're working with monkeys. So now, I want to give you an example with genomic editing, but specifically to a disease. So we can put it in words with something specific. So if you define Parkinson's disease, it's typically described as a movement disorder, and it's characterized by the loss of dopaminergic neurons in the substantia nigra and the presence of intracytoplasmic inclusions called Lewy bodies, and also these aggregates in the neurites. Those aggregates are mostly made of alpha-synuclein. Although that is the textbook story, Parkinson's disease is much more complex, has a number of different areas that are affected. Dorsal motor nucleus of the vagus, the pedunculopontine nucleus, the peripheral sympathetic ganglia are affected. And the Parkinson's disease symptoms reflects the complexity of the disease. You have patients that have problems with anxiety, patients that have problems with the heart, patients that have problems with constipation. So Parkinson's is more complex than just that nigrostriatal problem. What causes Parkinson's disease? Well, we have a pretty good idea of the mechanisms. In the last 5 years, we have learned a lot about what causes Parkinson's. We know that there is a role for inflammation, a role for oxidative stress, and this alpha-synuclein aggregation seems to have a very important role. Now, if you're asking me, what causes Parkinson's disease? I cannot tell you. We are all working towards this. We know there is, if you're getting older, your risk of having Parkinson's disease is there. If you have been exposed to toxins, if you have a car accident, you have brain damage, you have a higher risk of Parkinson's. And of course, the reason that we're here, genetics. There are several genes have been robustly associated with Parkinson's disease. But in Parkinson's disease, basically we say that it's 5% of the population of patients with Parkinson's carry this mutation. And in 2004, we learned about this particular gene, the LRRK2 G2019, LRRK2, that specifically the G2019S, that changed the perspective of why we're looking at genetics in Parkinson's, because this particular gene is the most frequent and has been found in sporadic and familial cases of Parkinson's disease. And in certain populations can be 30%. So this is an interesting target to analyze, huh? Now, what models we have today in Parkinson's disease in monkeys? Well, we have all monkeys. We have alpha-synuclein gene transfer, but not the fancy things that these guys have been talking. We're talking about you have your viral vector that's encoding for your gene of interest, and you put it inside the brain. And in the area of the brain that you place it, the gene is being overexpressed. That's the model that we're talking about. And then there are all these different ways they administer 6-hydroxydopamine. We have some attempts like cardiac dysautonomia models in Parkinson's disease that my lab has done, but by and large, all these models share one problem. We are always focusing in the dopamine and the substantia nigra. And that's not Parkinson's disease. And also they're mostly acute. So, sorry, I don't do history as our colleagues, mainly because we don't have much to tell you about the history of monkeys with transgenesis. we have a few publications. It's 15-year-old, basically, story that in the early 2000, we have GFP rhesus, and then marmoset later on in 2009, where lentiviral vector was put in the embryo and then was transferring to the host, and here you got a transgenic animal. In 2008, we have a Huntington's disease model where a CAG repeat, the Huntington repeat, was done the same way. And more recently, A53T alpha-synuclein cynomolgus monkey has been reported. Basically the same thing, not CRISPR. CRISPR are this just last year appear. PPR gamma and RAG1 in 2014 by New et al. Duchenne syndrome a month ago, well, I think a couple of weeks ago, and the Kang et al. With the X-linked adrenal hypoplasia congenita and hypogonadotropic hypogonadism, that's one knockout. So the field of monkey transgenesis is very young. And I think a lot of the progress is being made as we understand more about the human and non-human genome. We have better targets to put the effort into in a specific disease. And because we are learning how much is affecting the progress of treatments, the current models that we have. So I think it's interesting to remember that all these mutations are single point mutations. So those models that have the lentiviral vectors sound interesting, but they are expressing of the molecule, and is not the type of phenotype that a human has, with the exception of alpha-synuclein trisomies that are a handful of patients that have presented that. So I think that genomic editing can provide a solution. And again, if we think about the applications, let's go first to the models. And because this is so young, what I want to share with you is the considerations that we are having at Wisconsin when we're thinking about this development that we got funding to do, and we are in the process of creating these animals. As I mentioned to you, LAC2 seems to be to me, the most interesting gene because of how much this is in the world. It will be very interesting, I said, alpha-synuclein, but there are so many few patients that have it. So that's the reason we are targeting LAC2. The next thing is that you need to keep in mind, and this is a sobering thought for the people that works with other species. A rhesus macaque, the adulthood, it has 5 years. The gestation is 5.5 months, and they have one baby at a time. It's hard to make transgenic monkeys. Marmoset monkeys, or Callitrichus, they have a few advantages compared to a macaque for transgenesis or genomic editing. They can give you twins or triplets. They have, and most interesting, especially if you're working in Parkinson's, they have a shorter lifespan and they reach adulthood sooner. And if you're looking for the phenotype that is appear later in life, you need to think about what species you are using. So this is our approach. We have this marmosets. Of course, in monkeys, you have to have your donors and your hosts, and everybody has to be synchronized with the hormones, and it's a lot of work and a lot of monkeys. And I'm looking at Buddy Capuano, who is the attending veterinarian at the Wisconsin National Primate Center, and he knows how many animals we need to get these monkeys to breed. They are a lot. So we have these animals, and the idea is, well, I want to have these animals. This is what we are in the process of doing, in vitro fertilization of ICSI to have the cells. And one of the things I want to mention before, we do not know why many of the technologies that have been so successful in mice do not transfer well in non-human primates. We do have, for example, embryonic stem cells. Actually, Jamie Thompson developed embryonic stem cells in rhesus and marmosets in the 90s. So we had the stem cells, but when you try to put those cells into the zygote, it doesn't work very well. Into the embryo, it doesn't work very well. We don't know if it's because we need to work more on the methods, on the reagents, but it's not so efficacious. So this is the way that it seems to be most efficacious in monkeys. And we have in marmosets, part of the work that we are doing is we are also obtaining stem cells, and we have recently produced induced pluripotent stem cells from marmosets, and we are using them as platforms for the CRISPR-Cas9 strategy. And this is just to show you that we can take skin cells and make them into pluripotent stem cells. And I'm a neuroscientist. We can do basic neuronal differentiation in marmosets iPS cells, and we can make them into mature neurons. They express beta-3 tubulin, and we can make them dopaminergic, that they express tyrosine hydroxylase. And this is the work of my grad student, Scott Vermilea, just to show you that how we are targeting LRRK2 gene, and that we can we have confirmed that we are targeting the right loci. Now, I cannot show you the success yet. I can show you only that we are successful at targeting. But we are ready, in the same way that when we started thinking about this, about what gene, what species, as important as all that is how you evaluate the animals. These are monkeys. Every monkey is precious. Live a long life. I cannot outbreed a mutation that I don't like. I need to evaluate it and analyze it. That's the investment that you have to do when you're working with non-human primates. And this is to show you the analysis that we have done about how a marmoset monkey changes over time. And this is to remind you, a baby monkey is not a small monkey. Has different needs, has different neurobehavior, and you have to have tests that are appropriate for that age. And we are ready, when those animals are born, to evaluate them over time with all kinds of different tests, not only to observe the gene expression, but the overall health. We have developed neurobehavioral postnatal scales that did not exist before. That was part of our project. So when the babies are born, to evaluate how healthy they are. Not to say just, oh, they are developing normally. No, we are going to know what these animals are doing and how they are doing. We are looking at not only the motor tests that are the typical features of Parkinson's disease, but also at the non-motor tests to study the olfaction, to study cognition, to study gastrointestinal function. We also we are going to perform imaging to assess if the effect in the brain as peripheral nervous system. And as you may see, we have scheduled specific time points that are matching the evolution of the animal and trying to be the least invasive as we can, while maximizing the data that we are collecting. And why we're so interested in all those new motor symptoms? Well, first of all, these are monkeys. Every little bit is important, but because patients have all these other problems, and it seems that non-motor symptoms can predict Parkinson's disease. And if we identify when those motor symptoms appear, we could initiate pharmacological or lifestyle strategies or some kind of a strategy that can prevent the onset of the disease. This is one of the ways that we look at this as how to help with the therapy. So with that, let's think about how we can use genomic editing for therapies. Again, we're thinking about it from the point of view from someone that has Parkinson's. And with my colleague Sushan Shan, we have successfully done personalized stem cell therapy, meaning we had fibroblasts from a monkey that were transformed into dopaminergic neurons. Of course, it's never that easy, and put it back into the brain of the monkey. And the monkey has been Parkinsonian through a neurotoxin called MPTP. And we had found integration, and this is just to show you the personalized cell therapy approach. And now, what we have figured out is it works. No, we can replace the cells, and we are in the middle of a functional study to assess how much of the motor function can be recovered. But one of the things that we had figured out with this is the cell replacement is more than just playing a puzzle, and that host and donor have a role in graft efficacy. So it's not that you have these beautiful cells and you put it in the brain and the brain is perfectly healthy. There is a lot of mechanisms or things that are in play at this moment. And genomic edited monkeys that have the disease would be able to give us an answer if you put the healthy cells in a sick animal, if those healthy cells can survive over time and provide the relief that the patients need. Also, we have a chance, if I have a monkey that has the disease, can I genomic edit his sick cells and then put it back in the monkey? Personalized medicine, one step from the human. And one of the things that we are working also with Sushan is using genomic editing to incorporate into these stem cells that are dopaminergic a designer drug receptor, so we can modulate the activity of the cells and, for example, increase the release of dopamine or stop it, inhibit if the patient is having abnormal movements. Then the last thing I wanted to talk to you about, therapies in the germline. I think that this is part of the conversation that we need to keep going with the human genomic editing. You are aware that if you try to genomic edit me, it's not gonna work. Unless maybe if I have polyps and we inject some electroporate some plasmids like you were showing, Angelica. But at this moment, it's not possible. And I think genomic editing for therapy of the disease or for enhancement has to wait until this conversation is solved. So now the last two slides, I want to summarize some of my thoughts about these considerations when you are working with primates. First, you need to think before you're embarking in this travel that for rodents or zebrafish is well traveled. It's not that well for monkeys. You need to think first about disease-related issues. What mutation is the most valuable to invest the time and effort? The age of onset that the disease is going to have, and what symptoms have to this mutation may cause. Then you need to think about questions related to the species. What do we know about the genome, a lifespan, what developmental patterns that animal have? What I show you is for marmosets. It's different if you're working with rhesus monkeys. What housing needs these animals need? You cannot have it in single cages. Are you gonna have it in groups cages? How you are gonna make sure that they don't breed at the wrong time, things like that. Breeding demands. How many animals are you gonna have as hosts and as donors? How you can synchronize their cycles so your recipients are ready when you got this blastocyst, or how you're gonna solve this problem. Also, monkeys are outbred like humans. What is gonna be the impact of the individual genetic background? So probably at the end we'll finish with enough inbred animals that we do that, but how we're gonna solve this problem is something that we need to think about. Then the other thing I think is very important is the testing. The testing has to be, of these animals, has to be specific to the disease, the age, and the species. And then of course, all the genomic edited related issues. The methods for plasmid inoculation, the uniplex, all this, what are you gonna do, one gene or several genes, the random insertions and deletions, the target effects, the mosaicism, penetrance. We haven't talked about penetrance. And the reason I have this image here is because I think it's important to know your species genome. What you see here is the alpha-synuclein, that protein that is so interesting in the field of Parkinson's disease today, the residues 51 to 60. humans, like great apes and most of old world monkeys, have at the 53A locus an adenosine, unless humans have a mutation, that is, a threonine. Interestingly, most of New World monkeys, rats, mice, Galapagos turtles, they all have a threonine in that location. So if I try to do a mutation in that particular place, it's not gonna be useful. And we need to know this. We need to think about it. Also, why marmosets do not have Parkinson's? And that means there are other genes that are interacting with that one. Food for thought. So I think overall, when you're working with primates, it's with any species, but with primates in particular, you have to keep a what-if scenario and be ready with a clinical veterinarian support for any of the problems that can emerge. And I think also that we need to keep this conversation going. And overall, I just want to share with you some of the practices that a while ago we discussed with Jonathan Kimmelman, my colleague and friend from McGill, about what are the ethical basis for initiating first in human trials, but it's also with any research. You have to have internal validity, meaning that you need to know what is your hypothesis, power calculations, randomization, blinded treatment allocation. It's not just, oh, we cured this monkey. It doesn't work like that. External validity, why you are selecting that animal model, why you are choosing those outcome measures, what are your quality control measures. And when you are going to go to a clinical trial, there has to be a correspondence between the trial and the preclinical conditions. And the last, we need to curb our optimism and publication bias. We need to be critical provide critical review of preclinical data and do full publication of preclinical studies. And I think we as scientists, as reviewers, as journal editors, as grant funders, we need to be aware of it and be cautious and not penalize scientists that take their time or publish negative results. And with that, I want to thank you. And these are my colleagues. A lot of this work has been done with Scott Bermilea. He's a brilliant grad student in my lab. With my teammate, Tig Golos, and his group, Jenna Kropp, Greg Whips, and Michael Mayer. And I also want to thank Sue Shan and Jamie Townsend for advice. This will not be possible without the support of the Wisconsin National Primate Research Center and his director is John Levine, and who supports this initiative. And also, just a couple of things more. Mark Cookson from the NIH, who is always there when we need him for like 2 questions. Alex London and Jonathan Kimmelman challenging with ethical questions. Thank you very much.